All right. Well, thanks for the opportunity to moderate this talk. My name is Larry Kwong. I'm an associate professor at MD Anderson. I share a number of interests with Stephanie, which is, I guess, why I'm asked to moderate today. Our lab works, as Stephanie's does, on a lot of patient samples, looking at both the genomics and histopathology. I'm very excited to see her talk today. I've been told to encourage people to please interrupt the talk. You can do so by putting your questions into the chat or Q&A, and I will interrupt on your behalf politely. So without further ado, I'd like to introduce Stephanie Ressler, who is a professor at Heidelberg University Hospital. She got her PhD at the Max Planck Institute of Immunology and Epigenetics, and later did her postdoc at the NCI under Dr. Xin-Wei Wang. And now her group is focusing on identifying genetic and epigenetic alterations in both hepatocellular carcinoma and cholangiocarcinoma, and to use in vivo and in vitro models to look at mechanisms of liver cancer development and progression. Today, she'll be talking about morphomolecular characterization of early and progressed cholangiocarcinoma.
So thank you very much, Larry, for this very kind introduction, and thank you for having me and giving me the opportunity to present some of our data. And yeah, I'm not only going to present our own data, but also include some other groups' data, of course, to bring it all in context. And I'm looking forward to yeah, a lively discussion and interaction. So yeah, I'm also a member of ENSCA, and I'm really honored to be part of the molecular classification group within ENSCA. And so today I will mainly focus on different characteristics of cholangiocarcinoma. And so with this, just very briefly, we are mainly focusing at intrahepatic, perihilar, and distal cholangiocarcinoma. So yeah, we are separating the CCA groups into the anatomical localizations. And the frequency of these tumors may vary between different cohorts. So in general, the intrahepatic cholangiocarcinoma is the rarest type, followed by the distal, and then the perihilar is the most frequent type. Over the years, it appears that there is an increase in incidence and also mortality of cholangiocarcinoma. And yeah, here you can see some different numbers from different countries. And you can also see this is not always the same. This might be due to some data issues that have been provided or that were not recorded. But overall, in general, mostly the incidence and mortality rates were increasing. And so it is important to understand why is this the case, and how can CCA be prevented, how can it be detected, and maybe how can we treat CCA better to improve patient outcome. One important thing is the etiology, the different risk factors for CCA. And as you can see here on the left-hand side, the risk factors, they vary greatly. They range from liver cirrhosis, HPV infection, to very general things like diabetes, which is occurring very frequently. Alcoholic liver disease has been implicated in some studies. The data is not completely certain for alcoholic liver disease, but most likely there is also some association. And some of these factors, they are very frequently observed in specific cohorts. For example, liver fluke or primary sclerosing cholangitis. And some of these factors are also very rare, like Carollis disease or Lynch syndrome. So overall, there's a large number of different underlying etiologies for CCA, and this may also cause the high heterogeneity of CCA. And as you can see, there's also differences in association between intrahepatic CCA and extrahepatic CCA, and the different etiologies. And so, for example, liver cirrhosis is mainly associated with intrahepatic CCA, versus other factors like, for example, cholangiocarcinogenesis is more associated with extrahepatic CCA. So, there might be also differences in the anatomical localization of CCA. In a very early study for whole exome sequencing, there were the first clues given that also the mutations might differ between the different etiologies. So here shown is CCA without liver fluke, so CCA patients who didn't have liver fluke, or CCA patients who had liver fluke. And you can see that some mutations, for example, mutations in the BAP1 gene, they are more frequent in the non-liver fluke patients. In other cases, for example, P53 is higher in the liver fluke patients. The same is also true for SMART4 and GNAS. In contrast, IDH1 and IDH2 mutations, they are more frequently observed in the non-liver fluke CCA patients. So also the gene mutations, they vary between different cohorts with different etiologies. A larger study followed up, and in this study, there were similar findings observed. One example is the BAP1 gene that was also observed before. This one is more frequent in frequently lost or mutated in the fluke negative patients. In contrast, for example, P10 was in this study observed in the fluke positive patients. So, but what you can also see here is that still this picture is very heterogeneous. There are so many different mutations, and also there are a lot of patients where we don't find any mutations or any known driver mutations, where we don't understand how these tumors may have arisen. So there's a lot more to analyze, and it might not be all explained by genetic alterations and by these mutations. And so furthermore, it is important to integrate the data. So not to look only at the genetic alterations, but also at the epigenetic alterations. And so this is a very nice example where also the DNA methylation as a readout for the epigenetic changes was analyzed. And this was combined with gene expression, copy number, and also in addition, the mutations. So here there is different clusters that were identified. Cluster 1 shown here has mostly fluke positive patients. Cluster 2 was kind of mixed, whereas clusters 3 and 4 had mainly fluke negative patients. And you can nicely see here for the example of DNA methylation that there are great differences in some patterns of the DNA methylation, but also in the gene expression data, and as discussed before, also in the genetic alterations. And it is really also very striking that also the patient survival was different here in these different clusters. Also patient outcome is associated with different subtypes of the tumors and with the different etiology of these tumors. And so there is most likely a very strong link between etiology and the development of the cancer and also progression of the disease. So in this case, these 4 different clusters are plotted in the Kaplan-Meier curve. And you can see down here in panel C that this fluke negative cluster 4 had the best outcome. And this cluster contained mostly patients without fluke. So also just looking at the differences between the anatomic location of CCA, this is also giving a nice difference between the mutations in the patient groups. So we have the intrahepatic, perihilar, and distal cholangiocarcinoma patients, and you can see here that, for example, p53 mutations are more frequently observed in distal CCA. Also, SMART4 is more frequently observed in distal CCA, whereas IDH mutations are in this study exclusively found in intrahepatic cholangiocarcinoma, but also other studies find a much higher prevalence of IDH1 and IDH2 mutations in intrahepatic cholangiocarcinomas. So there might be also differences associated with the anatomic location. And right now people are more and more considering intrahepatic, perihilar, and distal cholangiocarcinoma as separate diseases, and are also more and more looking separately at these different cancers. So this is a summary of different mutations that have been identified, which are different between intrahepatic and extrahepatic CCA. And you can see that there is a long list of different genes, and the differences, they vary. Some differences are very clear, like for BAP1 and IDH1. For other factors like p53, it is not as clear. So there's also quite some numbers of intrahepatic CCA that have p53 mutation, and also in the extrahepatic. So it is very important to consider the anatomic location of CCA for molecular analysis and to understand these diseases. So in addition to these mutations and epigenetic changes, there is also more and more classification of tumors and subclassification based on the histomorphology. And so this is now also part of the WHO. And so the ICCA cases, they are further subdivided into small duct type ICCA and large duct type ICCA. And here are some examples shown from Liao et al., who very nicely also reported these different groups of ICCA. And I think I like this picture very much because you can still see that there's variability. So it's not black and white, but there are different small duct type ICCA and different large duct type ICCA. So it will be very important to further analyze these different morphological subtypes, and to better understand also the biology in this morphological growth pattern, and also to better understand how this might be used for treatment and for better understanding ICCA. So one thing that is suggested is that the ICCA subtype, also shown here, is arising from the interlobular zone of the intrahepatic bile ducts. And so this is a different type of cell of origin for ICCA, whereas the large duct type ICCA, they arise more from the peribiliary glands or have other precursors. So that maybe the cell of origin is different between small duct type and large duct type ICCA. In one of our studies, we performed.
Stephanie, can I interrupt you? Sorry about that. On your last slide.
Should I go back to the slide before?
Yeah, I just have a question about that. As you know, a lot of mouse studies have shown that cholangios can arise from hepatocytes. Is there evidence for that in humans as well?
This is a very good question. So in mice, it is clear that cholangiocarcinoma can also arise from hepatocytes. In humans, it is not very clear, at least not to my knowledge. There are some studies, of course, suggesting this, but there is also, and I didn't really go into this part, also the cholangiocarcinoma, the combined hepatocellular carcinomas. So there's also another subtype, and it is not very clear whether only CLC is arising, maybe also from stem cells, hepatic stem cells or progenitor cells. It is unclear whether CCA can also arise from mature hepatocytes. So I think in the community, people are more thinking that it is progenitor-like cells that can also have specific types of hepatocyte markers. At least from my feeling here, it is not so likely that really mature hepatocytes are turning into CCA. But it might be, and I think there's more research needed to really clarify in humans whether this is possible.
Is there any good approach to answering that question? Obviously, we can't do lineage tracing in humans. So what sort of options are there to address this question more?
Yes, of course, we cannot do experiments in humans. I think we can look into different hepatocyte markers and also maybe differences in the genomic DNA of hepatocytes versus cholangiocytes, non-coding areas maybe that are specific to hepatocyte differentiation. And maybe this is one way to support the hypothesis that also hepatocytes may give rise to CCA.
Yeah, I think cell of origin is really interesting. There happens to be a question here from Joanne, which is anything known on the IOV of small duct versus large duct type ICCA?
Sorry, can you repeat the question?
Yeah. Can you see the question on the Q&A?
Okay, I will.
Inter-observer variability.
Yes, yes, so I think there is a considerable inter-observer variability in small duct versus large duct ICCA classification. Also here in Heidelberg at the Pathology, there's a lot of discussion about how to classify some ICCAs that have structures of both and are not very clear really say this is a small duct versus large duct type ICCA. So some tumors, they have parts of both. And I think there should be more consensus in the community and more discussion about these cases that have features of both. It is not completely clear from the WHO how to classify those cases. Okay. So yeah, there's still more work to do, and I think also maybe imaging might help. So to use artificial intelligence in the future to depict more these differences, and definitely there is need for a better consensus on ICCA on the large duct type and small duct type ICCA. So if this is question is answered, yes, Joanne agreed. Thank you. So then I will continue with our study where we looked at 52 ICCA samples, and we performed gene mutation analysis, both exome-based and also panel-based sequencing. And we also performed DNA methylation analysis. And what is nice about DNA methylation analysis, there's also algorithms to calculate copy number variations and to also integrate this. And for data integration, we used the Medicom analysis for DNA methylation alterations that are underlying. So what we found is that there are different subgroups of ICCA patients, and shown on top is depicted the small duct type and large duct type for these ICCA. And you can see here that there is one group that has only small duct type ICCA. And these cases had a high frequency of IDH mutations. So therefore, we named this group the IDH group. And the other two groups we analyzed further. And what we found is that there is a degree of differences in methylation of these three groups. The group to the left had the least DNA methylation alterations. The middle one had intermediate, and the right one had high methylation alterations. And therefore, we named them L, M, and H groups. And in addition to the DNA methylation, also the copy number variations, they were also the least in the L group, intermediate in the M group, and the highest in the H group. So there are different molecular alterations in these four ICCA subgroups that we identified. And the IDH group had only small duct type ICCA cases. Not all of the cases had IDH mutation, but it might be that there's also an alternative or mechanism that is mimicking IDH, or that there's another mutation, not the hotspot mutation that we couldn't observe. And the other three groups, they were mixed with small duct type and large duct type, and they also had a different degree of mutation numbers. Interestingly, the IDH mutations, they were all hotspot mutations, and this is also observed in other tumor entities, and that was also observed in previous studies for CCA. So it is mainly the arginine 132 in IDH1 and arginine 172 in IDH2. And also the patient outcome was different in these 4 ICCA subgroups. The IDH subgroup had intermediate to good outcome, and the low alteration group, the L group, had the best outcome, versus the intermediate M and also the H group, they had the poorest outcome. So also the genomic and epigenetic alterations, they are associated with patient outcome. And yeah, there was also the TCGA study by Fashidfa et al. In Cell Reports, and also they identified this IDH mutations and specific epigenetic alterations in these patients. So this is a specific subgroup of ICCA patients, and this gives an opportunity to really use this molecular characteristic of ICCA for targeted therapy, and to better understand the etiology and the development of this IDH subgroup of patients. Just very briefly, what is this IDH mutation doing? IDH1 and also IDH2, if they are mutated at this hotspot arginine, they can lead to the conversion of alpha-ketoglutarate to 2-hydroxyglutarate, and this in turn is changing a lot of different factors within the cell. Most notably, it's also changing epigenetic factors, the histone demethylation and also DNA methylation. It's also involved in hypoxia, and so there is a wide range of alterations in IDH mutant tumors. What I find very interesting is also that this variety of alterations, also epigenetic alterations, is also causing a plasticity of the cells. And in animal studies, there was immune evasion identified in IDH mutant cancer models. And also it appears that there might be some tendency of resistance development if the IDH mutant protein or the IDH mutant gene is inhibited. So yeah, there is a lot more to analyze in these IDH mutants, ICCAs, and to better understand. But recently there was a clinical study. It's the CLARIT trial, and here they used IDH1 inhibition in IDH1 mutant ICCAs. So patients with IDH mutant CCA were enrolled in this study, and the inhibitor ivosetinib was given to the patients, or there was a placebo arm. And so what you can see here is that ivosetinib significantly improved patient overall survival. And yeah, this was a very positive outcome. You have to consider here that these were patients with very advanced tumors and very aggressive tumors. So overall, there was no cure. But yeah, I think it's a clinical trial going into the right direction. There might be also the necessity to perform other dual treatments in case of resistance. But I think it is clearly showing that there might be a benefit of really selecting patients with specific mutations and then treating those patients with specific inhibitors against these mutations, like it was done here in this IDH1 mutant trial. So in another study, we looked into PSC-related bile duct cancers. So PSC is one of the very strong risk factors for biliary tract cancer, BTC, and PSC patients. They have a greatly increased risk of BTC development. And what is, the cause is that PSC is leading to inflammation, fibrosis, and multifocal biliary strictures. And what is very bad for the patients is that patients are very frequent quite young and under 60 years of age. And this is a difference to most other CCA patients who mainly develop CCA at later times during their life. So there's a great need to survey PSC patients, and it is very difficult to diagnose CCA in the PSC patients. So it is important to better understand PSC-associated BTC and to identify better ways of screening patients and identify the patients that are developing CCA. In a multi-center study, we recruited PSC patients from several European, but also US-based centers. And there was 224 samples that we received. After QC and sequencing, we were left with 186 samples. And so this is the cohort of 174 patients with invasive carcinoma that were analyzed further. And what you can see also here is that the mean age was 48 years. So it's quite young actually compared to other BTC cohorts. And there were patients with ICCA, PCCA, CCA, and also gallbladder cancer, GBC. In addition to performing panel sequencing, there was also pathological analysis of the samples, and this was done by Benjamin Göppert mainly. And so he looked at the histology of these tumors. And what you can nicely see here is that there seems to be also differences in patient outcome in the different histological subtypes. And shown in the Kaplan-Meier curve, you can see that the solid tumors depicted here in red, they had the poorest outcome, whereas papillary tumors, so tumors with papillary growth, they had the best outcome. So I think it is very important to further characterize also the histology of the tumors and to better understand how these tumors differ and how this might be also translated into the clinic.
Stephanie, I have a question about
the
last slide about the histology. How often do you see intratumor heterogeneity, and how do you deal with those? Do you go with the most dominant histology type, or do you not see it that often?
Yes, this is a very good question, because tumor heterogeneity is, of course, a big issue. And so here we go with the dominant type usually, to make a decision. And of course, for this, it is good to have quite some areas of the tumor that can be analyzed, to have full slide H&E. To be able to really depict the tumor morphology, the histology, the histological type. Again, like with the small duct versus large duct type, there is some inter-observer variability, and definitely it would be good to have more consensus. But in general, I think most people are going with the dominant type within the tumor for the histology.
And how often do you see intratumor heterogeneity, or like for each tumor, are you looking at multiple slides from different regions, or does it generally just one slide?
Yeah, so in most studies, especially here with the PSC BTC study, we had only one H&E slide. So this is the limitation that, yeah, we could not have several areas of the tumor to analyze. Of course, yeah, in research settings, when tumor tissues are collected, with this purpose, it is different, but in reality, for most studies, I think we don't have yeah, the really all areas of the tumor. So we have to go with the dominant type within this section that we have.
That's very understandable.
Yeah, yeah. But of course, it would be really good to microdissect these tumors so that we have different histologies and to also study this further. There are several studies for the CLC tumors to really look into differences. So I think, yes, it would be really good to look more into tumor heterogeneity and to better understand these different yeah, areas of growth.
Oh, actually, I see a question.
Yes.
Says PSC and PBC, primary biliary cholangitis, share a lot of similar features. However, PSC may be linked to CCA, but not PBC. What do you think of the mechanisms?
Oh, this is a very good question. And so I think it is not very good understood. So yeah, also the differences between PSC and PBC, how, yeah, so why patients are developing PSC is not completely understood. So there are some genetic factors that are also associated with PSC development. But it is not very clear what are the molecular mechanisms behind PSC development. So I think for this, it has to be yeah, also studied the very early PSC development. How is PSC arising? And then, yeah, PSC, of course, is causing the strictures. It has to be also something with repair mechanisms, but the differences between PSC and PBC, I don't think that they're very well understood in terms of CCA development. And in terms of, yeah, also the genetic alterations that are occurring and that are needed for CCA development in these patients. So this would be a very interesting study if anyone has good cohorts to study these differences. Good. So what we identified in PSC BTC is that really this PSC etiology is also driving the mutational landscape of the tumors that are arising in PSC patients. So in the different anatomic localizations, ICCA, PCCA, DCCA, and also in GBC, we had quite similar mutation frequencies, and the mutations that were found were mainly P53, KRAS, and SMART4. And based on the study by Waddell et al., these mutations are typical for the DCCA subtype in non-PSC patients. And this was also confirmed by other studies that DCCA have mainly TP53 mutations, SMART4, and very little IDH mutations. And in this regard, in the PSC cohort, we did not observe any FGFR2 translocations, and also there was no BRCA1 or 2 deleterious mutation. And we also had just one ICCA patient with an IDH1 mutation, which is quite low frequency for this cohort of 174 patients. So this is suggesting that the PSC BTC is mainly of the extrahepatic type and similar to DCCA, and that the anatomic subgroup may not differ molecularly in PSC patients. And another finding that was more like a side finding in this cohort was that the PSC BTC patients, they very frequently had papillary precursor lesions, also called IPNBs. And so this frequency is higher than, at least in most cases in the literature. In the cohort of 174 patients with invasive carcinoma, 34 patients also had IPNB in the bile duct or also in the gallbladder ICPN. So it might be that this PSC etiology is also increasing the IPNB development, but further studies are needed to better characterize the frequency of IPNB in different studies. This was just a very interesting observation, and led us also to think about IPNB and other precursor lesions. So in the last part of the presentation, I will ask the question, what are the very first steps of cholangiocarcinogenesis? And so there are different precursor lesions that are known in CCA. One is the biliary intraepithelial neoplasia, BILI, and this is mainly observed outside the liver, extrahepatic. And then there are two different types of papillary precursor lesions, the IPNB, intraductal papillary neoplasia of the bile duct, and also the ITPN, intraductal tubulopapillary neoplasia of the bile duct. And all these are suggested to be precursors of the invasive CCA. So what we did is first that we wanted to really microdissect these precursor lesions, and therefore we used laser capture microdissection. We enriched the normal biliary epithelium, the baleen epithelium, and also the epithelium in invasive carcinoma. And for this, we used only 12 patients, but for each patient, we had the triplet set of these tissues. So the goal was to go mainly for the differences and to better understand baleen-associated CCA development. So we only had DCCA tumors in this study because baleen is mainly associated with distal CCA. So what we found is differences in the expression patterns of different microRNAs shown here. There was differences, of course, between the invasive carcinoma, and there was a gradient expression in baleen tissues, and the greatest difference from invasive carcinoma was to the normal bile duct epithelium. So one of the microRNAs that we followed up functionally was microRNA 451A, and this is gradually decreased via baleen to the invasive carcinoma. And what is interesting about MIR451 is that it decreased the invasiveness of tumor cell lines, And one downstream target that is directly affected by MIR451 is ATF2, which is also involved in migration. So by overexpressing MIR451A, ATF2 was decreased, and then migration was also decreased. So yeah, with this we could confirm some molecular mechanisms driven by microRNA expression, and that there is a transition between normal bile duct epithelium via B-lean to the invasive carcinoma. In the next study, we then focused on IPNB and ITPN to better understand how these are rising and whether these are really the precursors of invasive carcinoma, and what are the steps involved in invasive carcinoma development. So for this, we had a cohort of IPNB and ITPN associated CCA patients. The IPNB and ITPN were characterized by immunohistochemistry. And one thing that we observed was that ITPN and IPNB patients, they had relatively good overall survival. And also when we accounted for UICC staging in IPNB or ITPN associated CCA and non-IPNB, non-ITPN associated CCA patients, we observed that ITPN and ITPN are factors that are associated with better overall outcome of the patient. So what are the differences between IPNB and ITPN at the clinical level? So here we looked at different factors, and one thing that panned out was that the localization of ITPN is mainly intrahepatic, versus for IPNB it was mainly distal. And this was also significant in the cohort. In addition, also there was a tendency in the histology that there was in IPNB more in intestinal histology, versus in ITPN we didn't have any. However, these numbers may not be large enough to really depict the histology, and therefore it would be really interesting to have larger cohorts and to further characterize IPNB and ITPN. So yes.
Question real quick, I'm not very familiar with this. Are these surgically resected with curative intent?
Yes, so all of these cases were resection cases and with curative intent, yes, yeah. So UICC was mainly 2, some were also 3. We also had some UICC 1, of course, but not that many.
So how often do they relapse?
So with IPNB, ITPN, we didn't have a very long follow-up in terms of relapse, but it was also much better than the CCA patients without ITPN and IPNB, like with the overall survival. But yeah, for relapse, unfortunately, we don't have much data, because some of the patients, they do not come back to the center here in Heidelberg, and then follow-up is not so easy all the time. Yes, but this would be really interesting. So one idea is that IPNB and ITPN, maybe also resulting in earlier diagnosis of CCA, but it is not clear how this is associated. At least in our cohort, we didn't see large differences in the UICC staging at least not that, yeah, they were significant here in our case.
Very interesting.
Good. So coming to the molecular alterations, we performed panel sequencing of the IPNB and ITPN. And what you can see here is that overall ITPN had fewer mutations in the known drivers and in the hotspot areas, the typical tumor-associated genes. And IPNB had mainly the typical mutations of distal cholangiocarcinoma, definitely extrahepatic, with P53, SMART4, KRAS. And there was only one IDH mutation, and it was in IDH1 and in one of the ITPN cases. So this is also consistent with the anatomic localization that ITPN occurred mainly intrahepatic, and they seem to be more similar to the ICCA compared to IPNB. So there might be different precursor lesions, intrahepatic and extrahepatic. But ITPN is not very frequent, yeah, at least it's not reported to be very frequent. So it is still unclear if there are distinct precursor lesions for ICCA. In terms of the DNA methylation profiling, we performed MEDICOM analysis of the DNA methylation arrays. And what you can see here is that there are different underlying patterns in the MEDICOM analysis. And all the ITPN cases clustered together and had mainly the LMC4 subtype. And this is suggesting that they have the same cell of origin. Unfortunately, with this data, we don't have any reference map to really say what is this cell of origin. So I think it would be really interesting to look at the DNA methylation patterns of different fractions of cells, of progenitor cells, and intrahepatic bile duct areas of different types to really define these patterns. And this might also support the hypothesis of different cells of origin for intrahepatic and extrahepatic bile duct cancers. So in the further analysis, we compared pairwise the IPNB or ITPN with the invasive carcinoma of the same patients. So here in this plot, the patients are ordered in the same order. The order is based on the ITPN or IPNB on the left-hand side, and the invasive carcinoma that is associated is presented in the same order. And you can see here that some of the mutations, they have the same frequency and they are identical between IPNB and the invasive carcinoma, like P53 or KRAS mutation. And other mutations are more frequent in the invasive carcinoma, like for example, ROBO2 that is shown here. So we didn't find any ROBO2 mutation in ITPN or IPNB, but we found alterations in invasive carcinoma. So we believe that over time there is a transition from the non-neoplastic normal bile duct epithelium via the precursor to the invasive carcinoma. And there are different mutations that are arising only in the invasive carcinoma, and some are already early on in the precursor lesion already present. So with this, I'm coming to the conclusion. Overall, I think CCA is a very heterogeneous cancer type. It is heterogeneous in terms of the histology, of the molecular alterations, whether these are genetic or epigenetic. And it is really important to better characterize these tumors. In regard to the ICCAs, we were able to identify at least 4 different ICCA subgroups. They are also associated with patient outcome. and also associated with specific gene mutations like in the type of IDH. So for this, I think there might be more subtypes, and it is really important also to better understand the tumors that do not have any known mutation or any driver mutation, to better understand these epigenetically driven CCA, which are also associated sometimes with poor outcome for the patient. In terms of the precursor lesions, we studied mainly IPNB and ITPN, and found that they are different in their mutational profile, and based on the epigenetic profiles, they might have distinct cells of origin. In addition to studying the tumor, it is also important to better understand the etiology. As an example, PSC patients, they have a very high frequency of CCA development. And in this case, it might be that PSC-associated CCA mainly arise via the IPNB precursor. So it is important to better characterize also the precursor lesions, to better understand how the tumors are arising, and hopefully then patients can be screened and identified early on, and hopefully there will be better treatment in the future. So with this, I'm coming to the acknowledgements. I would like to thank Benjamin Göppert. He is a long-term collaborator of mine here in Heidelberg, and it was always fun discussing with him the science. I also would like to thank all the group members and Professor Schirmacher here in Heidelberg for his support. There was a lot of exchange with other people in Heidelberg, and also especially for the PSC cohort, we were collaborating with Trine Folseras and Tom Hemming-Carlsen in Oslo. So with this, I would like to thank everyone for the interest in this seminar. And yeah, if you have any questions, please feel free to ask.
Well, Stephanie, so much. Thanks so much for the talk. It was great. Before I get to the questions, I just want to remind everyone that these are recorded, and this video, as well as all the other ones, are available on the Cholangiocarcinoma Foundation YouTube page and also on their website. If you need the link to that, you can ask Reham. So there is a question here from George. IPNB seem to have a high fraction of ARID2 mutations, 7%. Do you have any comments about that?
Yes, this is a very interesting finding. This is what we observed in our cohort. We don't have a clear idea why we observed this number. I think to really state that ARID2 mutations are more frequent in IPNB, we would need to have higher numbers. So it was only 42 IPNBs that we analyzed here. But it would be, of course, interesting to see whether this is really true. And so unfortunately, we don't know why we observed this relatively high number.
I do have one more question. So you did mention in one of the slides that I think all of the IDH mutants had a small duct morphology. Is there a broader sense if there's any connection between mutations and the morphology type? Do we not know? And if it's not connected, what are the things that most likely contribute to the final morphology?
This is a very good question. We tried to find correlations between the histomorphology and the mutations, and so far we couldn't find any correlation. So at least in our cohort, in our samples that we have so far, maybe with the numbers not being big enough, because some of the mutations are not very frequent, we couldn't observe anything. Yeah, we don't know definitely, but at the moment, it doesn't look like that the mutation is really causing the histomorphology of the CCA. So I think that especially for tumors that are very heterogeneous, there's definitely also some epigenetic effect within these samples. Within these tumors, there's cell plasticity, and yeah, the tumor cells interact with the tumor microenvironment, and that this interaction is mainly causing the histomorphology. Yeah, the interaction with the tumor stroma might be one of the driving factors, but we don't have enough support to really say what are the factors causing these morphological differences. So I think it would be really interesting to further dissect these tumors, whether it's with laser capture microdissection or by some other technology, to better understand the differences in the tumor microenvironment. Maybe also single-cell RNA sequencing might help here to really better understand the differences between this histomorphology and also the in this case, gene expression patterns, or also the differences between the patients in gene expression.
Thanks. Yeah, I agree. I mean, there's so many different things which could influence, you know, potentially you could think of cell of origin, the person's diet, age, gender, what hormones they got. So yeah, there's a lot of things that could affect it. So I just was hoping there was a more simple, straightforward answer. If not, it's, well, yeah, I agree. It's something very interesting to follow up on. And one of the other questions related to this that I always kind of struggle with is to what degree these different morphologies represent different degrees of differentiation. So for example, a cancer like bladder cancer, you could kind of easily define it as basal or luminal, but it seems like it's much more complicated for cholangio. And I don't know if the word differentiation even really applies to these different morphologies. Do you have an perspective on that?
So there is definitely some differentiation that can be observed between the different morphologies. I think that there is an association between the cell of origin that you mentioned, that is also giving rise to different morphologies. And also, I think there's this effect of differentiation, whether this is really coming from the cell of origin or whether this is the tumor-specific de-differentiation. This has to be studied further, I think. So we don't really know this degree of different differentiation. Whether this, since we cannot really follow the tumor, but it's like a one-point picture that we get at the time of resection. This is a difficult question to really answer where it's coming from. But I think definitely the differentiation is a factor also in CCA.
Okay, I don't see any other questions, so thanks to everyone for attending, and of course, thanks to Dr. Ressler for her great talk.
Yes, thank you everyone for listening and for the very nice discussion, Larry. Thank you.